So the summary so far, we've talked about the structure and localization of cells, and in particular, it's important to remember the different types of cells. So we have myeloid cells, like macrophages and dendritic cells, and lymphoid cells, like IELs, or B and T cells that are found in the lamina propria and Peyer's patches. We talked about important antibodies such as IgA as well, and how it can help protect the epithelial layer. And now we'll talk about control of the microbiome. Okay, so I mentioned the microbiome at the top, but really it's the population of different microbes that live on and within the surfaces of our body. In the gut, one concept that's very important is called oral tolerance. So this is an experimental example, but mice that are fed ovalbumin will actually have a kind of tolerizing response. So if they're injected later with the same protein, instead of having a severe inflammatory reaction, this reaction will be fairly muted. And this is really important in the intestinal mucosa because the intestine is really unique in that it comes into contact with things that are good for the body, such as food, and as well as coming into contact with microbe and antigens, which can be good or bad. So the gut needs the ability to respond to the pathogens, but not respond to the things that are good. And this is also important for allergy, which we're not going to talk about today, but we'll be talking about in a couple lectures. So just keep it in mind for later. Okay, so the microbiota needs to be controlled. And this is done through the innate and adaptive immune system. And as I mentioned, this is really an important balance between control and tolerance. So the microbiota can actually have the ability to become pathogenic if not properly controlled. And this is partially mediated through detection via M cells and presentation to immune cells. So in this figure, we can see M cells giving antigen to dendritic cells. And then these dendritic cells or other cells such as T cells will then produce cytokines and signal to different epithelial cells. And this will induce them to produce things such as more mucus and antimicrobial peptides. And this helps keep the microbiome at a reasonable layer. So we don't want an overgrowth of bacteria, and we also don't want bacteria invading the epithelial layer. So this is kind of a circuit that helps the epithelium know when it needs to provide more of an immune response. So if pathogenic bacteria take hold, such as after antibiotic treatment, which will kill off some of the good bacteria, allowing pathogens to infect, this can lead to dysbiosis, which can also cause damage. So this is an example, a pretty well-known one. So like I mentioned, antibiotics will kill off many of the good or commensal bacteria. And because now there's more space, this allows C. diff, which is a pathogen, to come in and colonize the gut. So we see C. diff coming in and producing toxins, and this causes mucosal injury. And if the C. diff is not held in check, these toxins will leak into the lamina propria and cause an immune response, which will lead to an influx of neutrophils as well as red blood cells into the gut lumen. So this then, if it continues, can lead to the breakdown of connective tissue and the disintegration of the gut. So what's really important is we want to maintain the good bacteria while also preventing bad bacteria from taking hold and leading to dysbiosis. So the microbiome is actually very important for normal immune development. So we can actually experimentally use what are called germ-free mice, which are mice that have absolutely no microbiome. And in these mice, they actually have a reduced immune response. Their immune system does not develop properly. So from these experiments, we can see that the microbiota is really important for the normal development of the immune response. So these are a few examples of how that works. So first, we have some specific bacteria, such as segmented filamentous bacteria, or SFB, and some of these bacterial proteins will be presented and interact and actually lead to the development of certain subsets of immune cells. And in particular, the most well-known is SFB, leading to the development of Th17 cells. Then we also have different proteins, such as butyrate, which are produced by some Clostridium or Bacteroides species. And the detection of butyrate will actually lead the epithelium to produce TGF-beta, causing the development of more regulatory T cells, which will then secrete IL-10. So this mechanism helps establish a nice anti-inflammatory phenotype, so we don't get too much inflammation from the bacteria in the gut. And then there are some other different organisms that different ligands of these will lead to either development of some of those IELs, or innate lymphoid cells we talked about earlier, or detection of these bacteria by the epithelium will lead the epithelial cells to produce cytokines and different mediator proteins. And these will lead to the development of different cells within the immune system. And usually, if all of this is working properly, we'll have a nice balance between cells that are able to mount an immune response if needed, without having cells that are going to be attacking the host and causing too much inflammation. So this figure shows a really good example of the inflammatory response when it gets out of control. So on the right here, we can see normal tissue, and on the left, we can see tissue in a patient with colitis. And you can see just how much thicker and inflamed that tissue is in the colitis patient. And this is due really to a very inflammatory response to the normal microbiota. So in this case, the patient is not able to form a proper balanced response. So it's mounting in a response too much to the microbiota, causing more inflammation and disease. Okay, so in summary then, the mucosa forms a unique barrier that interacts with the outside world. So we mostly talked about the gut, but there are other areas in the body, such as the nasal or vaginal mucosal surfaces, that also come into contact with the outside world and also have their own set of microbiota. There are many immune cells that reside within the mucosa, and this is really important since the mucosa is in contact with the outside world. We need to make sure that our immune system is controlling what gets through that layer and how it responds. So these immune cells can actually be within the epithelium, as we saw with the IELs, or in specific regions, such as the Peyer's patches. And they can also be scattered throughout the mucosa, like they would be in the lamina propria. So we have some innate immune cells that we talked about, such as dendritic cells and macrophages. And these cells are important because they help bridge the gap between the luminal side and the apical side of the epithelium. So we can see that these cells will take up microbial and food antigens, transport them across the epithelium, and present it to T and B cells. And when there's an anti-inflammatory environment, this antigen presentation typically leads more to tolerance, as we mentioned. So this supports an anti-inflammatory phenotype. So we're not seeing a large inflammatory response or disease if the things that the gut is coming into contact to are not pathogens. So we also see a robust adaptive response in the mucosa, and this includes IgA secretion, which we saw is secreted across the epithelium, and is important for binding and transport of different antigens. We also saw effector T cells, which are developed through the lymph nodes and Peyer's patches in the gut and help make an immune response to these antigens. And we also have regulatory T cells, which I've mentioned a few times and are very important to help control the inflammation. And finally, we talked about the microbiome as an important modulator of the immune system. So the microbiome provides a set of bacterial antigens that are not trying to infect the body. So the immune system almost trains against these antigens. So it can develop different T cell subsets or innate lymphoid cell subsets, and it will help the immune system not mount too strong of an inflammatory response. So overall, I hope this has helped tie together a few concepts from previous lectures you guys have had, and as well as pointing out the importance of the mucosal immune system. So thank you.